Blood is something we all have running through our veins, right? But the way medical professionals actually analyze what's floating around inside it, well, it can honestly feel a little bit like pure magic. So today, we're going to play the role of lab detectives. We're pulling back the curtain on a very specific and highly visual testing method that transforms a seemingly cryptic biological puzzle into a perfectly readable map. But before we get into the heavy mechanics of clinical tests, let's just step back and ask a really fundamental question. How in the world do scientists physically see the hidden differences in our blood composition? We all know blood isn't just some uniform red liquid. It is literally packed with different proteins and molecules. But to diagnose conditions, we absolutely have to have a way to separate and identify those incredibly tiny components visually. So, take a look at these pink smudges on the test strips. Now, to the untrained eye, sure, they might just seem like random ink blots, but they are actually a precise, highly readable sequence. That is, if you know the scientific code. Every single horizontal placement of those pink lines, it's telling a distinct, real-life biological story about the blood sample that was provided. All right, let's dive right into this pink band mystery and decode exactly what it is we're looking at here. The specific test we're looking at is called Alkaline Cellulose Acetate Hemoglobin Electrophoresis. Try saying that 3 times fast. I know, it sounds incredibly intimidating, but if we break it down, it tells us exactly what's going on. Basically, a special strip made of cellulose acetate is placed in an alkaline pH environment, and that environment allows the different types of hemoglobins to physically separate out into distinct, visible bands. Okay, so we know what the test is called, but let's talk about the actual mechanics of this migration. Like, how do these bands actually move across the strip? Think of this whole process like a miniature racetrack. The hemoglobins start their journey over at the negative cathode. Then, that alkaline pH environment we just mentioned, it gives these proteins a net negative charge. And because opposites attract, that negative charge actively propels them forward, rushing toward the positive anode. So, the absolutely crucial point here is understanding exactly why some of these runners are faster than others. In this biological race, hemoglobin A is our undisputed track star. It's the fastest runner simply because that alkaline pH gives it a much greater net negative charge compared to the rest of the pack. That stronger negative charge pushes it aggressively across the finish line, landing it way farther toward the positive anode than any of its competitors. Now that we've got a handle on how this microscopic race actually works, it is time to decode the hemoglobin sequence itself. Scientists actually use a very predictable order to identify every single runner on that track. And here's your ultimate cheat code. A2, C, E, then S, D, G, followed by F, A, and finally Bart's and H. This simplified little memory aid is really the practical core of interpreting these tests. Searing this sequence into your mind will literally let you read almost any strip out there. It maps out the exact order of the bands from the starting line all the way to the finish line. So A2, C, and E are your slowest runners, hanging in the back. Then you've got S, D, and G. Then comes F, followed by our extremely fast runner, hemoglobin A, and pushing at the very front of the pack are Bart's and H. Building on that cheat code, you can see the complete migration sequence mapped out right here, all the way from the starting origin, that negative cathode, straight to the positive anode. You can see exactly how the hemoglobins line up in their specific lanes. It's incredibly straightforward. If a band pops up in a certain zone, you simply reference this sequence map to identify exactly which hemoglobin type is hanging out in your patient's blood. Let's actually see how this builds into real-world interpretation, okay? Because to understand an abnormal test, we obviously have to establish a solid baseline first. A perfectly normal sample is going to feature a massive, dominant hemoglobin A band running way ahead toward the anode. And trailing behind it, much closer to where it started at the origin, you're going to find a very small, faint hemoglobin A2 band. Returning to our actual photograph from the lab, you can see this baseline perfectly illustrated. Just take a look at most of these individual lanes. You clearly see a really strong, distant pink band, right? That is our fast-moving hemoglobin A. And trailing far behind it, closer to the starting line, is that much smaller, fainter hemoglobin A2 band. It perfectly matches the sequence map we just talked about. But just when you think you have completely cracked the code of these pink bands, well, we have to talk about the co-migration complication. Yep, it turns out some hemoglobins act a bit like imposters. Here is the major twist. You actually cannot identify everything purely from this single alkaline strip. Why? Because certain hemoglobins co-migrate. Notice how hemoglobins A2, C, E, and O Arab will all bunch up in the exact same region. Similarly, hemoglobins S, D, and G migrate right alongside each other too. Because they run at the exact same speed in this specific alkaline environment, they essentially stack up and look like one single thicker band. Because of this whole co-migration complication, our work as lab detectives isn't quite done if an abnormal band suddenly appears in one of those bunched up zones. A good detective has to rely on specific follow-up methods to confirm the true identity of the hemoglobin. They might run an acid electrophoresis instead of an alkaline one, or they might use HPLC, capillary electrophoresis, or even go straight for direct molecular confirmation. These extra steps are absolutely crucial to figure out exactly which imposter is hiding inside that pink band. So where does that leave us? Well, the alkaline strip we explored today is a brilliant and beautifully visual starting point. It takes an invisible biological world and maps it out right before our eyes in vibrant pink. But knowing about those co-migrating imposters, that should leave you wonderfully curious. If a single strip only tells half the story, what other hidden secrets are waiting to be decoded in our blood? It really reminds us that human biology is incredibly complex, and it takes a whole arsenal of fascinating scientific tests to paint the complete picture of what makes us who we are.